The microscope, an instrument that forever changed humanity's understanding of the natural world, began its journey from simple curiosities to advanced scientific tools that now help us explore the deepest secrets of life. The desire to see what the naked eye could not dates back to ancient times, when early civilizations experimented with magnification using crude lenses or polished crystals. Even in ancient Assyria, around 700 BCE, evidence suggests the use of transparent crystals to slightly enlarge images, although their understanding of optics was limited. In Rome, by the 1st century CE, philosophers like Seneca the Younger noted how objects looked larger when viewed through a glass globe filled with water. These early experiments hinted at the potential of magnification, but did not lead to practical instruments. It wasn't until the late 13th century that the first reading stones, convex lenses made of glass, were developed and used by monks and scholars to read manuscripts. These were primarily handheld and laid flat over text to magnify them, but they were still a far cry from what would become a microscope. The turning point in magnification came in the late 16th and early 17th centuries in Europe, during a period of great scientific exploration. In the Netherlands, around 1590, spectacle makers Hans and Zacharias Janssen are often credited with building the first compound microscope. It used 2 lenses in a tube to magnify small objects. While their invention was rudimentary and its magnification limited, estimated around 3x to 10x, it was revolutionary for its time. However, the design did not gain immediate traction, as optical quality was poor and practical applications had yet to be realized. Nonetheless, it planted a critical seed. In the early 1600s, Galileo Galilei, the famed Italian astronomer, developed his own version of a compound microscope, calling it the occhiolino, or little eye. Galileo's version incorporated a convex and concave lens system and achieved better magnification. Though known primarily for his work with telescopes, Galileo's foray into microscopy showed how similar the principles of lenses applied in both instruments. In 1665, the microscope made a giant leap forward when English scientist Robert Hooke published his groundbreaking book, Micrographia. Using a compound microscope that he had refined, Hooke observed and illustrated a variety of tiny objects, including the intricate structure of feathers, snowflakes, and fleas. Most famously, he looked at a thin slice of cork and saw that it was made up of tiny, box-like structures which he called cells, coining a term that would later become fundamental in biology. Hooke's observations amazed the scientific community and the general public, and Micrographia became a bestseller, helping to popularize the microscope as both a scientific tool and a source of wonder. Almost simultaneously, another figure emerged who would push the microscope even further: Antonie van Leeuwenhoek, a Dutch textile merchant with a passion for lens making. Leeuwenhoek, without formal scientific training, ground and polished his own lenses to an incredible degree of precision. He created single-lens microscopes that, despite their simplicity, had magnifying powers far beyond the compound microscopes of his day, up to 270x. Using his tiny instruments, he became the first person to observe and describe single-celled organisms, including bacteria, protoa, and sperm cells. He referred to them as animalcules, or little animals. Leeuwenhoek communicated his findings through letters to the Royal Society in London, documenting a microbial world that had never been seen before. His meticulous observations laid the foundation for microbiology. Despite these breakthroughs, progress in microscopy stalled somewhat in the 18th century. Compound microscopes continued to suffer from poor image clarity due to optical aberrations. Many scientists preferred Leeuwenhoek's simple yet powerful single-lens microscopes. It wasn't until the 19th century that significant advances in optics and manufacturing revived interest in compound microscopes. One major improvement came from the work of Joseph Jackson Lister, who in the 1820s developed a method to reduce spherical and chromatic aberrations by using combinations of lenses spaced at precise distances. This innovation greatly improved image resolution and laid the groundwork for modern optical microscope design. Around the same time, Carl Zeiss, a German optical instrument maker, began producing high-quality microscopes in the 1840s. Zeiss's partnership with physicist Ernst Abbe in the 1860s proved to be pivotal. Abbe developed the mathematical theory of image formation in the microscope and established formulas for lens design. He also introduced the concept of numerical aperture and resolution limits. Their collaboration transformed microscopy from an art into a precise science. Zeiss also worked with Otto Schott, who developed new types of optical glass, further enhancing lens quality. By the end of the 19th century, the compound light microscope had become a powerful and reliable tool used widely in biology and medicine. In the early 20th century, scientists pushed the limits of light microscopy, eventually realizing that visible light imposed a fundamental boundary on resolution. To see even smaller structures, a new approach was needed. This led to the development of the electron microscope. In 1931, German physicist Ernst Ruska and electrical engineer Max Knoll built the first transmission electron microscope, which used beams of electrons instead of light to create images. Electrons have much shorter wavelengths than visible light, allowing microscopes to achieve far greater resolution. Ruska's electron microscope could magnify objects up to 400x in early designs, and later versions achieved magnifications of over 1 million times. The invention revolutionized materials science, virology, and cellular biology. Ruska would go on to win the Nobel Prize in Physics in 1986 for his contributions. In 1937, Manfred von Arden created the first scanning electron microscope, which allowed scientists to view the surface topography of samples in stunning detail. Unlike TEM, which required thin samples, SEM bounced electrons off the surface, building a three-dimensional image line by line. SEM and TEM together expanded the field of microscopy into realms previously considered unimaginable. Simultaneously, the fluorescence microscope was developed, using specific wavelengths of light to excite fluorescent molecules in samples. This technique allowed scientists to target and highlight specific cellular components, leading to revolutionary insights in molecular biology and medicine. The advent of confocal microscopy in the 1950s and 60s, credited largely to Marvin Minsky, added optical sectioning, enabling scientists to take sharp images of samples at different depths and reconstruct three-dimensional structures. In the 1980s and 1990s, innovations like two-photon excitation and spinning disk confocal systems made it possible to image live cells and tissues in real time without causing damage. The digital revolution of the late 20th and early 21st centuries introduced a new era in microscopy. Traditional eyepieces were increasingly replaced by high-resolution cameras and computer screens. Digital image processing allowed for real-time enhancement, measurement, and analysis. New algorithms enabled advanced 3D reconstruction and automated sample scanning. Super-resolution microscopy emerged in the early 2000s, breaking the theoretical limits of optical resolution. Techniques such as STED, PA-LEM, and STORM allowed scientists to visualize individual molecules inside cells at nanometer resolution, once thought impossible with visible light. These breakthroughs earned researchers such as Stefan Hell, Eric Betzig, and William Warner the 2014 Nobel Prize in Chemistry. In parallel, scanning probe microscopes, including the atomic force microscope and scanning tunneling microscope, emerged. These tools didn't use lenses or light, but instead felt the surface of a sample with an ultrafine probe. Developed in the 1980s, STM allowed scientists to image and manipulate individual atoms for the first time, while FM-enabled imaging of biological samples in near-native conditions, even in liquid. These techniques opened entirely new frontiers in nanotechnology, chemistry, and material sciences. Today, microscopes are incredibly diverse and specialized. There are light microscopes used in classrooms and advanced systems that combine confocal, fluorescence, and super-resolution methods in one platform. Electron microscopes are now common in major research labs and hospitals. Cryo-electron microscopy, a technique that earned its developers the Nobel Prize in Chemistry in 2017, allows imaging of biological molecules in frozen, near-native states, revealing their structure in exquisite detail. Artificial intelligence and machine learning are now being integrated into microscope systems to automate image analysis, pattern recognition, and even sample targeting. Meanwhile, portable and smartphone-based microscopes are bringing diagnostic power to remote and underserved regions around the world. From ancient lenses to quantum-level imaging, the microscope has undergone a remarkable evolution. It transformed from a curious toy into a cornerstone of modern science. It gave us our first glimpse of cells, bacteria, viruses, DNA processes, and atomic structures. It enabled countless discoveries in medicine, biology, material science, and engineering. The microscope is not just a tool, it is a window into the fabric of life, one that continues to expand our understanding of the universe with every new innovation. As technology advances, so does the power of this incredible instrument. Future developments may allow us to view even smaller structures, perhaps even quantum phenomena, or map the neural networks of entire brains in unprecedented detail. The story of the microscope is far from over. It is a continuous journey of seeing the unseen, driven by human curiosity and the relentless desire to understand the world in all its complexity.